So we're talking about the group of bacteria that are known as Enterobacteriaceae, and remember they have a certain characteristics that apply to them. So they are, I'll just repeat it here real fast. So they are gram-negative bacilli. They ferment glucose, so I'm just going to write glucose positive, indicating they're positive for glucose fermentation. They're oxidase negative, and then they convert nitrate to nitrite. So those are the physical and biochemical characteristics that allow you to say you have an Enterobacteriaceae member. There's quite a few members to this group, but notice the only one that we're going to really focus on, just because we're limited on time, is Escherichia. And so that's the E of E. coli, and so we're just going to be calling it that, but I'm just realizing what that stands for. This group of Enterobacteriaceae, first we're going to talk about that coliform group. And so remember, this is a subcategory within this Enterobacteriaceae group. And so coliforms are going to have the added characteristics. So they have all of the things that I listed above, plus they ferment lactose. So coliforms ferment lactose. And so then just realize that non-coliforms are not going to ferment lactose. So that's the primary difference. And then coliforms are also going to be present in the intestinal tract of many animals and humans. And so because of that, they are found in the soil and on plants and on decaying vegetation, and they can be found in water supplies, lakes, ponds, rivers, etc. And we use the presence of coliforms in our water supply to indicate how contaminated it is. And so this is something that's done in sewage treatment plants. And so before the water gets released back out, has the coliform count been reduced to sufficient levels to deem it safe to be released. So like I said here, we're just going to talk about E. coli. And so that's what we're going to do. So E. coli can cause a variety of different diseases. But remember a couple things. The first thing, everyone has E. coli in their gut. So we're not saying that all E. coli is bad. It's a really important bacterial species in the microbiome within the gut. And so what we're interested in is a couple things. So we've used these terms before, opportunistic infections, I'm just going to abbreviate that, and then also certain pathogenic strains. So that term we've used previously to describe the variation that exists within a specific microorganism. So we can talk about strains of the influenza virus or strains of E. coli in this particular example. We've previously seen this for strains of group A Streptococcus, also called Streptococcus pyogenes or Staphylococcus aureus as well. And so there's that word again, virulent strains. The strains that have the ability to cause disease are going to be able to do so because they have those extra bits of instructions, those extra pieces of DNA called plasmids. And so it's on those plasmids where there can be genes that encode for or provide the information to allow the bacteria to actually cause disease. And so one of the more common manifestations of disease by E. coli is gastroenteritis. So this can be caused by a variety of different organisms, but E. coli is a bacterial cause of this. And what allows these strains to actually cause gastroenteritis is the ability to make and release toxins. So these are some terms we've seen previously, and the definitions are really important to kind of make sure you're learning about. And so again, an exotoxin is a toxin made by and released from a living bacteria. Okay, so exotoxins versus endotoxins is something we talked about when we were talking about infectious disease. And so remember, endotoxins is that structural molecule found in the outer membrane of all gram-negative bacteria. It's called lipid A or LPS. And so E. coli is a gram-negative bacteria. And so yes, it does have endotoxin. All gram-negative do. But it also has the ability to make and release this toxin that as the bacteria enter your gut, it can release this toxic compound that can disrupt the physiology there. And so that's what's going to lead to a lot of the problems with these particular strains. And so exotoxins are toxins made by and released from living bacteria. Enterotoxins, there's that prefix again that relates to the gastrointestinal tract. So these are going to be exotoxins that target the gut or GI tract or digestive tract or whatever you call it. So that's probably the most common way that we see this bacteria, but there's some other ways it can as well. So urinary tract infections, it can also get into the blood and cause disruption, so we call it septicemia. It can affect the central nervous system. This is in a certain population that we see. So as we said, the virulence factor is often on plasmids, and there's a variety of strains. And so these strains are frequently going to be identified by these antigens. So again, O is for unique antigens on the outer membrane. The K, although it doesn't start the same, it's for a capsular antigens. And then flagella are going to be H antigens. And so I used the example previously, O157, H7, which we're going to talk about momentarily. And so that's talking about a specific molecule on the outer membrane and a specific molecule on the flagella that can identify this unique strain that we know can cause disease within humans. So we're going to talk about E. coli. Before we do that, we've already talked about that it's going to frequently target the gut. And so there's two disease manifestations that we want to differentiate. So diarrheal disease, which we all know what that is, but we do want to compare that to dysentery. And the primary difference that invasion, damage. So diarrheal disease is simply described by what you already know, which is a liquidy, watery stool. Dysentery is going to have that, but there's some additional things as well. There's some indication that the bacteria are invading beyond the gut, and so that could be blood in the stool or pus in the stool or something like that. So invasion and or damage. And so obviously dysentery is going to be a more significant progression of this gastrointestinal disease. So E. coli can cause either or potentially both of these two types of gastrointestinal diseases. And so this isn't even all of the different strains, but we're just going to talk about 3 different types or categories of bacteria, or more specifically E. coli. And this is based on their ability to either make toxins. And again, the toxins we're talking about are enterotoxins, as we saw in the previous slide. And then the ability to make toxins are going to affect their pathogenicity. So the first, they go by abbreviations, but the names are really helpful. So we have that prefix entero, and I put over here, remember what that entero means relating to the GI tract. So enteropathogenic, so a pathogen of the gut. And so this is probably the least invasive or least pathogenic form. And so there's a certain population that is most at risk. So we know that infants are more prone to gastrointestinal, and that's mostly because, remember what we talked about, they're still developing the microbiome and still developing immune responses, we'll say. So there's kind of a couple things that are working against them. If an enteropathogenic strain of E. coli is entered into the gut, because of the fewer numbers and maybe the less diverse microbiome that's present, it's easier for that enteropathogenic bacteria to actually establish itself, and that could be coupled with, or it's can be either or both of these things working together with one another. If the immune system still hasn't been challenged as much, it's not as capable as fighting off things. So we see in infants these enteropathogenic strains that can lead to fever, diarrhea, vomiting, nausea. And so, but again, this is a diarrheal disease, so still problematic if this is your child and you're concerned about it. And what you're mostly concerned about is, again, based on what we've seen, dehydration that can lead to bigger things. So that's what we have to be concerned about here. This is kind of ratcheting up the pathogenicity or the virulence a little bit. So here we have, again, starts with entero, but here we have toxigenic. So that looks like toxin, and when we see genic, that's like generating or creation of. So we're really talking about enterotoxin generating is what that literally means. And so this is a bacteria that you can be exposed to through the consumption of contaminated things. And so this is often why individuals get sick when they travel. They're going to be exposed to strains of enterotoxigenic E. coli that they haven't encountered before, and so their immune system has no mechanisms in place really to fight it quickly, and so you can develop a diarrheal disease. And so again, these are enterotoxins that are going to lead to that change in the physiology in the gut. Again, still just a diarrheal disease. This last one is obviously the most significant and most severe. It's called enterohemorrhagic. And so we still have entero, but here we're talking about hemorrhaging. So that's indicating invasion or damage. And so while this may start as a diarrheal disease, what we're talking about is the possibility of it developing into a dysentery disease. And so here's that strain that I mentioned, E. coli 015787. And one thing that it can do above and beyond the diarrhea is it can lead to an inflammation of the colon. So itis is inflammation of, and so colitis. And so we're talking about damage, invasion/damage to that colon, which would be indicative of a greater concern than just a diarrheal disease. So we want to talk about this specific strain. And so again, notice how it's numbered. We talked about these are antigens on the surface, outer membrane versus flagella. And so there's a variety of disease-causing strains, but the reason why we want to talk about this one is it's going to be the most common or prevalent strain of E. coli in developed countries. So this is what we see in the United States, and we see this in Europe. And so it starts off as gastroenteritis, a diarrheal disease, and it could progress to dysentery. And actually, if it's, again, dysentery indicates invasion or damage to the intestine, there can be invasion beyond the gut and lead to other things that we'll talk about. And so one of the most serious intestinal issues that we can talk about with E. coli is what we saw before, hemorrhagic colitis. And so what that looks like in terms of a patient is blood in the stool. It's still in the diarrhea. And this might seem like not an important issue, but we'll see in other diarrheal diseases or other dysentery diseases where there's blood in the stool, few leukocytes. So there's not white blood cells in the stool. That's going to differ from when we get to Shigella momentarily. And this is a fancy word for without fever. So this is talking about what's going on in the intestine, but there's another concern that we have that it can actually lead to a really bad systemic infection that can lead to what's called hemolytic uremic syndrome. And so I'll talk about that in just a second, but before I do that, I want to talk about what's down here. So these E. coli O157H7, many strains produce what's called Shiga-like toxin. And this is like Shigella toxin. So it's not a very original naming. That toxin in Shigella is called Shiga toxin. So they put Shiga-like toxin here. So these two bacteria are pretty similar. So this toxin can cause this inflammation and invasion that we see within the gut. And so that's going to be a really important reason as to why this bacteria can cause this problem. But then there's additionally hemolysins. So we've seen that again. So hemo is related to blood, and lysins is to lyse or to break open. So the ability to produce hemolysins are going to start to affect the blood cells within the blood, and that leads to a type of anemia because the blood cells are breaking down. So it's hemolytic anemia. So that's the hemolytic part. But as you're breaking down those blood cells, you get a disruption. You get these fragments of blood cells that, as they're filtered out in the kidneys, can cause issues at the kidney level. And so you can start to have filtration issues, and kidneys can start to fail, potentially leading to the needing a kidney transplant over time. So it can get quite serious, and eventually death as well. So I'm not saying that each time this strain can progress to each of these, but these are ways in which this particular strain can progress, and we see it in patients. And so we want to be aware of what this bacteria can do. In terms of how people are exposed, it's through consumptions of contaminated food. And so there's been a variety of different sources of this, whether it has been linked to ground beef at different restaurants or different juices or this and that. And so you can search for E. coli outbreaks, and you'll see all sorts of recalls associated with it. One last thing, and this is really just reinforcing some things that we've seen previously. Remember bacteriophage. There can be lytic and there can be lysogenic. Lysogenic phage are those that use the lysogenic cycle, and so they integrate into the host bacterial cell genome. And so we actually know that this this Shiga-like toxin has been inserted into the bacterial genome through the insertion of a lysogenic phage. We call those a prophage. And so that's how it actually acquired it. So it's that that process of moving of genes between different bacteria through a virus, and we learned that's called transduction. So here's an example of that actual process. It's not just a theoretical thing, but genes can be transmitted between bacteria, a donor and a recipient, through the activity of viruses, of bacteriophage, and this is one example. So the toxin is encoded by a lysogenic phage that's inserted. These hemolysins are from the virulence plasmids that we talked about earlier as well. So there's a lot going on here, from the clinical manifestations to how it's transmitted to the virulence factors that are acting in a way that leads to the clinical manifestations. So the last thing in regards to treatment, I think I've touched upon it, but I'm going to spend a little bit more time on it. So it really depends upon how aggressive the disease is. So as we've seen previously, if it is simply a diarrheal disease, then basically it's really just treating the dehydration if it's present. But you're not going to do much to the actual bacteria. If There are severe cases where the bacteria has gotten into the bloodstream or there's signs of invasion. So if it's getting to this colitis or definitely the hemolytic uremic syndrome, indicating that there are toxins in the blood that are affecting the blood cells, well, that's a different story, and likely antibiotics would be warranted. But remember that antibiotics, not only will they disrupt the bacteria that's making you sick, that's the goal, but it can disrupt the gut physiology as well. There's trillions of bacterial species in the gut. And so that's really the balance that is trying to be kind of assessed when it comes to treatment. If it's a diarrhea disease and you treat with antibiotics right away, you could further disrupt the gut physiology, and that might not help the outcome. So often diarrheal diseases are going to be allowed to let them take their course, but really focused on making sure the patient isn't dehydrated. Okay. So what we're going to do is move on to, these are still in the Enterobacteriaceae family, but these are known as true enteric pathogens. And so these are true pathogens, meaning that unlike the other, like E. coli, which we call an opportunistic infection, these guys aren't supposed to be present in us. And so if they are there, they can definitely cause disease. And so we've already touched upon Shigella, but we'll add Salmonella and Yersinia to that as well. So each of these is a genus that we're going to talk about, and we'll go through them in the order in which they're presented there. So Shigella is going to be first. Remember that because these are enterics, they are all gram-negative rods or bacilli, and they are glucose positive, oxidase negative, and they reduce nitrate to nitrite. These guys are not coliforms, so they do not ferment lactose. So those are the characteristics that we have for all three of these guys. When I say all three, Shigella, Salmonella, and Yersinia that we're going to get to. So there's a variety of species of Shigella, but I don't necessarily care about the individual types. So we're just learning them as the genus Shigella. So the different species are going to be dependent upon where you are on the planet, and there are some different kind of severity of diseases, but we're just going to learn them as Shigella kind of collectively. All of them lead to the same disease, which is dysentery. So we've already defined that. This can also be called shigellosis, related to the bacteria that causes it. And so in this case, being a dysentery disease, blood in the feces, but then also notice mucus and pus containing leukocytes, which is different than what we saw for the E. coli version of this. And there is that Shiga toxin I was talking about before. So this is really similar to how E. coli O157H7 or enterohemorrhagic E. coli is going to act. So upon ingestion of these bacteria, so either enterohemorrhagic E. coli or Shigella, they each produce a toxin. They're really similar. They go by similar names. So this one has Shiga toxin. The E. coli has Shiga-like toxin. They are both enterotoxins, meaning, again, I'm trying to say this multiple times to really reinforce it. Living bacteria make and release toxin. And so that definition is that of what we call an exotoxin. But since this exotoxin is going to target the gut, then we can call that an enterotoxin. And that's indeed what we do call it. And so this disease is only found within humans. And so it is kind of a fecal-oral route of transmission. It's in the gut, so it's passed out in the feces of infected individuals. And so then it's really about sanitation and hygiene and hand washing and about proper food preparation and all those things that we know are important. So that's the route of transmission, and that's really how we can prevent this as well. In terms of treatment, it's really similar to what we see in that enterohemorrhagic E. coli. It's really a question of how severe is it. So it's that balance of fluids for preventing dehydration, and then if there is indication that the bacteria are causing some damage to the gut lining, then antibiotics may be warranted. So this shows kind of what happens when one of these Shigella is in the gut. And so again, this would have been contaminated food or water that is ingested. And what we're seeing here are those cells that are lining the gut. And so Shigella have the ability to bind to and attach to those cells lining the intestinal tract, so that we see that there. And these actually are going to enter the cell through triggering of endocytosis. So we've seen this in other types of microorganisms that we studied, like viruses that trigger entry into the host cell. So some bacteria can do this as well, and that's what Shigella indeed does. Once it enters, I'm going to put an extra step here. It's going to seem kind of random for the moment, but you'll understand why in a little bit. So during endocytosis, what you get is the bacteria in a food vesicle. And so there's another step here where that food vesicle is broken down. And so then you have the bacteria in the cytoplasm or the cytosol of this intestinal cell. And what it's going to do there is then the Shigella can multiply within that individual cell. So instead of having one bacteria present, you can have many of them. Not only do they multiply within the cell, but they use a technique that not all bacteria can do, but they can polymerize a protein that's called actin. And basically actin are proteins that are important in the cell skeleton, and so it can kind of allow them to form like kind of railroad tracks of sorts, so they can move along them and get pushed and enter neighboring cells. And the reason why that's important is when bacteria leave a cell, it is there where the immune system can attack them. So for example, a phagocytic cell, if is here, could come and eat that and destroy it. And so by staying inside of host cells, it helps avoid the immune system and allows the bacteria to spread and the disease to potentially spread, avoiding those immune defenses. Over time, that can lead to the destruction of that cell. And so you can then, here it says a mucosal abscess. So remember the enterotoxin called shigatoxin that these cells can make, and it can cause damage, and so the cells can die. And then the bacteria can then invade neighboring cells, or they could potentially get into the bloodstream. The good news is Shigella doesn't do well in the bloodstream. And so phagocytic cells, like neutrophils or monocytes, are going to quickly recognize, phagocytize, and destroy. So again, those steps are ingest and digest. And that's indeed what we see. What we're going to see momentarily is how Salmonella progresses in the gut. And so it's really similar, but there are 4 important differences that I'm asking you to note. And so we'll get to those in just a second, but you can think of it as kind of a game of spot the differences, like you have 2 pictures side by side, and you need to find what's different about each of the pictures. And so we'll do that momentarily when we actually get through Salmonella, but I'm just planting the seed now so you're aware that's what you're going to want to be able to do. So moving on to Salmonella, still this gram-negative enteric, so it has the characteristics of Salmonella as well. But this one is not only in humans, but found in a variety of different organisms. So it's in the intestine and therefore the feces of many birds, reptiles, and mammals. And so because of that, individuals who handle these types of organisms are going to be prone to being exposed to salmonella. But because these are important, at least bird and bird products like poultry and eggs, come from birds, they're a common source of exposure to salmonella as well. So there are different types, but again, we're just going to talk about salmonella. We are going to talk about this one. Some would argue it's a strain, some call it a species, but there is a specific manifestation that we can see of salmonella in humans called typhoid fever. So we'll talk about that one in just a little bit. But this too is going to be ingested and get into the gut, and so it's going to progress. And so you can see this looks really similar to what we saw for Shigella, but this is how salmonella can progress. So same sort of thing. Here's a salmonella that's been ingested, and I'll give you a hint here. Does this, I'm going to say gut cell, look the same? Hopefully you notice, I'll jump back and forth. When we look at this cell versus that cell, they don't look exactly the same. And that's because of where in the gut that we're talking about. So notice one difference. So one difference is where within the gut. So small intestine versus the colon, which is going to be the large intestine. Here we have the same sort of outcome of attaching to the cell. It's going to trigger endocytosis, so that's similar. Notice in this case, it shows the food vesicle. So I mentioned the food vesicle in the last one, but in this case, salmonella is actually going to divide or multiply within the food vesicle itself. So that's a second one. Where divide within the cell. So is it in the cytosol or is it within the food vesicle? That differs. And these salmonella can lead to a similar sort of outcome where the cell can die. So you can then have the bacteria entering the gut, or they can be entering neighboring cells. This particular bacteria, salmonella, is missing the ability to polymerize actin and spread, so we don't see that in this particular species. And when bacteria move into the blood, bacteremia, while there are some strains that are not able to progress well into the blood, there is one strain that we want to talk about that's called Salmonella typhi, which is the causative agent of typhoid fever. So Shigella does not do well in the bloodstream, but there is one strain or one species of Salmonella that does. And so the ability to survive within the blood can be different as well. So those are those four things that you can kind of pick out differences on between these two. So what we want to talk about is what happens when these bacteria of Salmonella typhi enter the blood, and that can lead to what's called typhoid fever. So typhoid fever is only going to be in humans, and so this particular strain is not present in birds or reptiles or other mammals. And so it is exclusive to humans, and so that means then that typhoid fever is going to be transmitted through the contamination of food or water that is contaminated. So again, this is really much driven by sanitation and hygiene that's going to minimize individuals' exposure. And so those bacteria, as we saw, are going to pass through the intestine, get into the bloodstream. So that's just what we saw before. But then once it's in the bloodstream, it can travel to different parts of the body, the liver, the spleen, the bone marrow. And the gallbladder is one of the important areas because it's the gallbladder that has important connections to the intestine that's necessary for for fat digestion and absorption. And so bacteria that have seeded the gallbladder can continually get into the gut And what you have is the ability to have a recurrent infection. And not only that, you can have someone that's classified as potentially over time, we've seen this for different things, asymptomatic carrier. So even though the bacteria are in their gallbladder, they are going to be releasing bacteria in their feces. And so if they are not doing a good job of hand washing, then they can be infecting a bunch of other people who can get sick. So in history, you can look up an individual called Typhoid Mary. She was a food preparer in the city of New York for some fairly wealthy, affluent individuals, and she was an asymptomatic carrier of typhoid. And so she kept infecting different families, and they would fire her. And anyways, you can read the story if you want. But nonetheless, it illustrates the importance of this. There are parts of the world where this disease and this bacteria is endemic. So remember what that means. It means that the disease occurs there at some frequency. And so if you're traveling to that part of the world, there is a vaccine that gives you temporary protection. I think it's about 6 months or something like that, 6 months protection. So you can take an oral vaccine, or you can, to help protect your gut, or you can get, I think there's a shot as well. Okay, so the last true pathogen of enterics is the genus Yersinia. There are 3 medically important species. There's probably more, but there's 3 listed here. These first 2 do really similar things. So enterocolitica and pseudotuberculosis. So these are both, notice, contaminated food and water affects the intestinal tract. So you get diseases much like we talked about, diarrheal disease, like we saw for E. coli, Shigella, Salmonella. Depending upon which of these 2 you have, it might be more inflammatory or less so. But since we've kind of already seen gastrointestinal diseases, we're not going to talk much about them. So other than knowing these 2 can lead to diseases, that's about it. So what we really want to focus on, I don't like the wording of that, not intestinal disease. So it is a member of the Enterobacteriaceae family, so it is an enteric, but it's not going to be transmitted through food and water and affect the gut. And so Yersinia pestis is the causative agent of plague. And there's several manifestations, so bubonic plague, pneumonic plague, there's and we'll talk about those in just a little bit. So this is looking at the life cycle of Yersinia pestis. And so this is a zoonotic disease. And there is, I already had it there, vector transmission. So the way that this bacteria is moving from the animal reservoir to the humans is through this insect, this biting arthropod, a flea. So there is a lot of arrows that show how this bacteria can move between populations. So in this case, you have a rodent that's the reservoir host, and so you can have fleas feeding on other rodents, and so it can spread that way. Obviously, fleas don't have to feed exclusively on rodents, but they can interact with other mammals as well. And so they can be what are called amplifying hosts that actually get sick and potentially die from it. But prior to doing that, you can have another flea bite this and then bite other things, etc. So you have this natural life cycle that's going on of the flea, the bacteria, and rodents and other animals in nature. But obviously, you can have fleas that interact with humans as well. And so that flea bite is one way that this can be transmitted. So that's kind of the most common way. It is possible through direct contact with an infected mouse as well, but fleas are going to be usually the route that we're focused on. So we're going to talk about bubonic plague And so this is where the bacteria can get into the tissues, so through that bite. And so without going into too much physiology, but there's something called the lymphatic system that's going to be collecting fluid from all of our tissues. And that fluid called lymph is going to be traveling through these lymph vessels, and there are collections of lymph nodes that are really important in immune response. And so we can get inflamed lymph nodes from all sorts of infections, but what you get during a bubonic plague infection is a really, really inflamed and large lymph node. It's called a bubo. So that's why it's called bubonic plague. And so that's a pretty clear disease manifestation, these buboes that can appear, and lymph nodes are localized to different areas, like axillary in the armpit and in the groin as well. But the bacteria, because it's in this lymphatic fluid, it could get in the bloodstream, it could spread, and if it gets and establishes itself in the lungs, then that's when it can be called pneumonic plague. And so if there's bacteria in the lungs, excuse me, then you can have transmission of this that's called airborne. And so through respiratory droplets that we're very familiar with coronavirus, but there are bacterial diseases like this as well. So bubonic plague wouldn't be transmitted person to person, but pneumonic plague could be. But it is possible for an individual who starts off with bubonic plague to develop pneumonic plague, or vice versa. If the bacteria are in you, it can potentially disseminate. So these are the three forms that I've kind of mentioned, bubonic plague. And so we have headache and chills. I'm not sure what I was trying to say there. Would have been fever. But most importantly is the formation of those swollen lymph nodes. And so here are some that are shown there. And so those are really, really large. In regards in addition to bubonic or buboes forming, if the bacteria in the blood, you can have what's called septicemic plague. And so we've seen that term before where bacteria or toxins are in the blood, so kind of a blood poisoning of sorts. And so when the bacteria are in the blood, it can lead to a certain pattern, a certain rash that forms. And so these kind of spotted appearance that you can see here is resultant from that endotoxin leading to again, disseminated intravascular coagulation. We saw this, sorry, I wrote that wrong. That should be a C, not a V. Disseminated intravascular coagulation. So we saw something similar to this pattern in meningococcus that can cause septicemia as well. Once this process has started to occur, where you're having this disruption of blood vessels and blood flow, that disruption can lead to the inability of nutrients and oxygen to get to extremities, and you can get the blackening of that tissue. So you likely have heard this, but this was also called the Black Death because of the necrosis that can occur, that tissue death and damage that's occurring that results from the presence of those bacteria. And then lastly, we have pneumonic plague. We mentioned that is going to be specific to the lungs. And also, what's important to know is person-to-person transmission. So bubonic plague and septicemic plague are not going to be transmitted through those mechanisms. We don't see a lot of plague cases, but just to show you how it's played out in the U.S. over the different recent years, here's the Four Corners area, and so most of the cases are going to be localized in that area. So if you end up being a healthcare provider in any of these Four Corner states, you may be witness to some plague cases. So again, this is showing how many of them over the course of the recent years. So it's never a lot, but the concern is that if someone does have any of these clinical manifestations of plague, it can progress really rapidly and could lead to death. And so treatment is going to be really important to diagnose someone quickly. So in terms of diagnosis, as I mentioned, it's got to be fast because of the potential of progressing to death. So depending upon which type those buboes are going to be really important, an important sign or symptom of that. But then beyond that, if it's in the bloodstream, it's really doing a blood sample and culturing, which takes some time. And so if plague is suspected, then antibiotics are going to be started regardless of if you know or not, but if it's suspected. One other thing I guess I could mention, a known flea bite in these areas, since we know that's how it's transmitted as well. So we've seen different types of antibiotics, and so this is going to be a cocktail of antibiotics. So we've talked about the different categories of antibiotics. So protein synthesis inhibitor for gentamicin, and the quinolones are the nucleic acid synthesis inhibitors.